All right, here we are with Tallan Black. Tallan Black is actually my colleague and friend. We actually work in the same lab under Dr. Robert Laprairie. So welcome to the, I guess, kind of YouTube channel podcast hybrid that I run, Tallan Black. And I guess what I want to talk about first is, I like your research. Your research projects are really cool. I've always taken keen interest. And could you talk about your master's research? Could we go over that first? Your master's research project where you're bonking animals on the head and looking at basically replicating traumatic brain injuries and the expression of cannabinoid type 1 receptor.
Yeah, honestly, when you just introduced me, I got a little shivery. Like that was really cool. So thanks for that.
Of course.
And thanks for appreciating my research. It's always cool to know your colleagues kind of back you and are interested in what you're doing, although it's vastly different from what you do.
Yeah.
But yeah, so my master's research. So like you said, I investigate traumatic brain injury, and I specifically dwell on kind of moderate to mild traumatic brain injury, a little bit more serious than what one might call a concussion, but not kind of the serious potential life-threatening injury. So within that, I'm looking at quantifying, or I looked at quantifying the endocannabinoid system. And you've highlighted the endocannabinoid system before, but to give everybody a quick refresher, you have two main receptors in the endocannabinoid system, the CB1 or cannabinoid 1 receptor and the cannabinoid 2 receptor. And both of them work a little bit differently in the central nervous system because the CB1 receptor, it acts mostly on neurons. And then we have the CB2 receptor, which is mostly involved in immune function. Oh my God. Immune function. And it's present on microglia. Now, like when you're thinking about the architecture of the brain, it's really important to keep in mind that there's a lot of things going on. So like if you think about the neuroanatomy, you've got like different levels of neurons stretching from kind of like deep within, brainstem, your spinal cord, and then that like reach out to frontal, like your peripheral lobe. So that's kind of the cortex portion that we talk about all the time. And all of those neurons, they communicate in vastly different ways. And in order to support those, we have kind of glial cells. And so that's where those glial cells help support, feed, clean up. They're just like, they're the Robin to the Batman.
I like that.
Yeah. And so anyways, the importance is to investigate kind of both components of the endocannabinoid system, right? Because there's a lot to it. But what I focused on specifically was the CB1 quantification. So that's the quantification in neurons. And what I did was I modulated the endocannabinoid system in brain injury with the administration of tetrahydrocannabidiol, which is, THC. Everyone knows about it, so I don't think I have to explain that too deeply, but it's a partial agonist at both CB1 and CB2. So that's why I kind of dwell on CB2 a little bit, but, a master's project is so long, so I've focused on the CB1. But within that, I kind of examined the sex differences between males and females, and I looked at how one administration of a lower dose, so 1 milligram per kilogram, direct kind of an hour after brain injury, whether or not that helped or hindered recovery. So I looked at it in rat models and I did a weight drop, and it was a modified weight drop model because I'm an athlete at heart, and it's important to get those rotational and accelerational components of a brain injury into a moderate to mild brain injury. You're laughing at me.
So basically what you're trying to do is you're trying to mimic a brain injury that somebody might get playing football or hockey.
100%.
Right.
Yeah, I'm trying to cover all the bases and kind of incorporate some of what is really truly what motivates me in research.
I like that. That's yeah. Great.
But yeah, so I did that kind of injury, and I gave them a brain injury, and then I administered THC, and then I looked at how it implicated their motor function. And then I looked at how it implicated their some more intricate behavioral functions, so, such as like working memory and then like anxiety, which are common post-traumatic symptoms. So important to look at. And then I also looked at the molecular components. So that's kind of what you're, I think, the most interested in, maybe, or just like a little bit of everything.
Yeah, everything is good. I mean, I really like this project. I remember we talked, off camera audio. We've talked about this quite a bit, your research project. So I'm interested in it generally. And
Cool.
when you administered CB1, agonists like THC, did you looked at how they recovered like right after the injury? So you'd administered THC right after the injury. And then you also looked at like motor function. So what was your study design? I guess let me ask you that. Talk about that.
Yeah, great question. So I gave them a brain injury. I administered THC 1 hour after, and then I measured their performance after 7 days. And then I collected their tissues to examine kind of any lasting molecular functions. Because typically moderate to or mild to moderate brain injuries, the symptoms will persist for 1 to 2 weeks for about 70% of cases. They can persist longer than that for the remaining 30, but I wanted to kind of keep that main pathophysiological and molecular cascade kind of in mind when I planned that time point. So I collected tissues at that 7-day time point, and then I examined CB1 concentration using Western blot, which is just quantifying that protein after a wild process, which you could probably do your own podcast on explaining.
Yeah, because most of the time Western blot doesn't even work, right? You have to troubleshoot so much BS.
Honestly, I was really lucky. Like Anna Maria, another colleague of ours, she worked so hard.
For like a year, and nothing worked.
And she was so patient and persistent. And like, honestly, whenever something doesn't work in the lab for me, I always try to channel my like inner Anna Maria stoic scientist, because it's a real thing.
It is so
the struggles.
If you're listening, Anna Maria, we love you and care about you. And
yeah,
thanks for your dedication to the Western blots.
So much, thank you. So I did Western blots, and then I did a cytokine and chemokine analysis, which is the quantification of inflammatory biomarkers and those molecules that signal throughout an injury process and recovery process. And then that was kind of it. And then I looked at everything and said, what the heck? What's going on?
What does it mean?
What does it mean?
Do you have any? Okay, so I know we talked about this a little bit. I asked you what was the overall arching results of your master's project, and you were like, it's complicated.
Yeah, like,
is there a general consensus? Like, basically, I guess I'll ask you is like, you obviously defended your master's and you went through this with your committee, and you had to have some sort of conclusion. And what was that general conclusion?
Well, it was, I think, okay, just to preface this, okay? So I did, like, this experiment was drawn out over such a long period because of COVID. And so I had like a big 6-month gap where I just like most people, you're at home, couldn't really do much. So I wrote my thesis and defended my thesis before I was actually done the molecular component, which was hilarious in itself. But now what I can tell you about my research is that there is a significant sex difference between males and females and how they react.
Let's definitely hear about that. That sounds great. I like that.
Like, so we, from a clinical setting, that has been reported, but it's been complicated because, you know, you get socioeconomic factors, and women are often caregivers, and so they kind of feel that pressure to return to a caregiver role, and so it's difficult to kind of glean that from any clinical research. So looking at the endocannabinoid system from my perspective, we knew that there were likely going to be sex differences, but we really just weren't sure what. And what it showed was that males actually had like a more robust behavioral reaction to the brain injury, whereas females just seemed to be a bit more resilient.
Wow.
And then on the other side, though, females had a significant change in their CB1 concentrations. And it kind of reversed itself between TBI with vehicle versus TBI with THC. So I had like this significant increase in CB1 receptor concentration in the TBI with THC, but a decrease in the TBI with vehicle, which was so interesting. And then on top of that, the cytokine profiles of males persisted as well. So females, I got an interesting result. I actually, like, I can go into it if you want me to, but like males, their cytokine profiles differed more significantly than females and more consistently. Whereas females had some funky stuff going on, but the important part is that like there is no research on chemokines and cytokines modulated like by the endocannabinoid system. Like it is slim pickings if you want to delve into that research. There's it's coming out, but there's a lot of unknown. So it's like its own, like if you want to look at how cannabinoids affect inflammatory mediation, like holy smokes, there is so much room for exploration there.
That's interesting.
Yeah. Mm-hmm.
Would you, okay, so you said there was this large difference, sex difference between males and females. Females seem to upregulate CB1 receptor, it seemed like. Would you, is this like, has anyone, okay, so like, what's your hypothesis? Is it hormonal? Is it progesterone, estrogen mediated, or is it something structurally different within the brain? Do you know? Have you thought about it?
Oh, I have combed through
What's
this
your
part.
hypothesis? I'm curious. What do you think, knowing more about the field than I do?
Yeah, so like going back to TBI, it seems that research indicates that females are more resilient to brain injury from a molecular component, which seems to be associated with primarily estrogen, but also some progesterone.
That's interesting.
But it isn't necessarily super robust. Like there is like ovarectomized when they like remove ovaries and inject males with estrogen and inject females with estrogen, and like yes, it has been shown, but it's not always consistent. So there seems to be a lot more to that story. But interestingly, there is a robust difference between like young rodents versus kind of mid-age rodents. And it seems to be that hormonal fluctuation throughout puberty or like adolescence in that rat life. So if there was actually one paper that I dug into by Makaziak et al. She's kind of one of the researchers that really helped me with this project. And one of her students showed that THC in adolescent rats before injury, after injury, they had similar reactions behaviorally, males and females. So I kind of adopted that into mine, and afterwards different behavioral reactions. And so when I dug into the research, there was loads of stuff indicating that like TBI reactions differ from that adolescent to mid-age, and the speculation is that it is hormonally related. but it's complicated in a human population. So
Totally.
like, it's just,
Yeah.
yeah.
The only thing I do know regarding hormones and CB1 receptors, there was a paper that came out of a Spanish research group in like 2012 or 13, and they showed that there is that hormone binding site on cannabinoid type 1 receptor. There's a pregnenolone binding site, which is a hormone, as you know, and it binds to CB1 receptor, and it actually acts at one of these allosteric modulatory sites, not the orthosteric site. So I think, I don't know, like, I guess bringing in my bias, it could be something like maybe hormones like estrogen and progesterone have a higher binding affinity than testosterone at that allosteric site. Maybe it's a better allosteric modulator. I don't
Well,
know.
that's interesting because females as a whole seem to have increased CB1 concentrations versus males, just in general across the board in both animal and clinical populations. And once again, it is like puberty dependent. So it's cool stuff. And like there's been a little bit of work in kind of the stress department there. So like pre-adolescent stress, and then kind of receptor concentration. And yeah, it's like females are different, and it's because we are different. We have other things going on. Like imagine.
Yeah, totally. I think the concentration dependent sex difference of CB1 is interesting. Is there anything known about CB2 in terms of sex differences concentration wise? Is it the same or not really?
I haven't dug into it as much. So that's like thanks for bringing that up. Like I'm gonna just write myself a little note there.
It's a research question, yeah.
Yeah, it really is. In terms of research in clinical perspectives, like CBD, which does act at the CB2 receptor, is kind of used the most frequently. But it like I'm sure you've reviewed, CBD is a little bit more complicated in its pharmacology. And so like,
yes.
its activity at the orphan receptors and other nuclear receptors, like that seems to be where some more of that is coming from, especially from a TBI perspective. So yeah, but I don't know specifically about the sex differences. So like, thanks for giving me that little bug, the research bug.
In the research world, we're always trying to figure out things and pass around research ideas. And I think a lot of times, we get caught in our own bubbles. I get caught in my molecular docking crap, and maybe you get caught in your animal stuff. And then when we talk to collaborators outside of our area, it's when we actually get ideas because we get perspective. And I think that's something interesting in research.
Man, it's so real. And I think that's part of the reason why my friend Elsa and I started Knowledge by Proxy. And that's part of the reason why I think we've been missing conferences so much, is because like you get stuck in your own bubble. And then like when you conference properly, like when you go and you like listen to like every single talk, even though it might not relate to you, and you just like you try to just bend your brain a little bit, that's when you start to get like these weird snippets of inspiration. Yeah, so kudos for us coming up with a new question.
Totally. All right, so that gives us some conclusive information about your master's project. I think your PhD project's even cooler. And I think due to how important this research question is, it will take you the full time of your PhD to figure this out.
Oh my God, I know.
I think you're going from one cool animal research project in cannabis to another even more intense research project in cannabis. Give us a quick overview of this PhD project. It is so cool.
Asher, thanks. I appreciate that. Yeah, so it is a really exciting project. So I should call it like a 4-part series. but it really is, but it's a PhD project, right? So that's like a lot of work, I've heard. I've heard from a couple friends, couple sources.
I love
Yeah,
it.
so I'm going to be I am examining maternal cannabis exposure, and I'm doing a pharmacokinetic analysis because I want to see the distributions of cannabinoids in maternal tissues as well as fetal tissues. And then I'm also going to be kind of taking that forward from a behavioral standpoint and examining the effects on different behavior subtypes in, once again, rats. So it's it's full on, and it's like combining a couple of things that I'm interested in, like pharmacology, sweet. Let's learn how to use a big complicated mass spec and do liquid chromatography, um and uh and then let's like just do such a comprehensive experiment that takes freaking ages.
And let's hear about how do you get both mice and rats high? Tell us about
It's my favorite part of it. So I get to hotbox rats. It's really fun though. We have a system from La Jolla. Note La Jolla, but they have provided us with this pretty cool setup where it's like 4 boxes. You can Google it too. 4 boxes, and you can put smoke through them, and they literally sit in this hot box of cannabis smoke. But you can use them on to vaporize alcohol. You can vaporize meth, and you can vaporize oils. So like cannabis oils. So you can do like a couple. And nicotine. Yeah. But you can do a couple different things. So it's cool. But we do cannabis in them, and it's neat.
And you guys are doing cannabis flower or cannabis like oil?
We are doing cannabis flower. Yeah.
And
So, you
and
go.
when you oil them, so I do want to get into your experimental designs. You have a certain amount of groups. You probably have like mice or rats that are pregnant, and you have them within different gestational periods of being pregnant, and you have some that are not pregnant. Can you give us the quickest sort of experimental design you can of your groups? I'm curious about this.
Totally. So specifically, I'm smoking them from gestational day 6 until gestational day 20, and I'm going to do the tissue collection at both first day, so GD6, and then GD20. And the reason for that is because I want an acute time point and then a chronic time point, so we can see that bioaccumulation. From there, it starts at GD6, gestational day 6, because cannabinoids have been found to interfere with embryonic implantation. So it's just like breeding rats is already a bit of a struggle, let me tell you from experience. So we don't need to make our lives any harder than it already is. But important to note that. And then I am treating them every single day with cannabis.
Wow.
And in terms of kind of control groups, we are working with THC, we're working with CBD, and we're working with just a control. And then from a smoke side, we're working with a high CBD flower, and then a high THC flower, and then an air control.
I didn't realize you had that much going on. You have a lot of different groups going on. Holy crap.
That's
a lot, Tallan.
It's mind-boggling. Like it feels like I'm doing like a large-scale Sudoku every single minute of the day.
Oh my god. And then you have like N of 6, and what's the N on that for animals?
For groups. So it's N of 8 for each treatment group, each time point. It's huge. It's huge.
Massive study. Jesus Christ.
Yeah, it's huge. Mm-hmm.
Whew. And then so you do all this animal work, which is already a lot, and then you have all your post cannabis analysis. And you said you were looking at cannabinoid tissue distribution.
It's a mouthful.
And how many different tissues are you looking at?
Everything on the buffalo, let me tell
Jesus
you.
Christ, oh my God.
No, but that's the beauty of our lab, is that there's numerous people versed in numerous different things. So
Right.
for me specifically, I'm looking at brain, maternal plasma, and then maternal liver, ideally. Okay, you know, like that's the goal. But then I also am examining placental and fetal brain.
And that's what's important. That's cool.
Yeah,
Because then you can
I
see the concentration of cannabinoids in the placenta, in the placenta, and in the offspring's brain to see how much of that permeated the placenta barrier, right? And that's
totally.
really cool.
Yeah, it's exciting. It's something. Yes, it's something.
That's awesome. And then you're doing some behavioral tests too. Can you give us a brief
Totally.
overview of the behavioral tests you run?
Yeah, so like right now I'm kind of optimizing some social interaction.
Oh,
Um, yeah, just because like social interaction is important from like a number of different angles.
yeah.
But specifically, if we're looking at it from kind of like a maternal cannabis exposure side, there was a huge study that came out of Ontario, actually, and I can't remember the name of the author for the life of me, but I'll give it to you so
Okay.
you can post it in the show notes.
Will do.
But this large scale study that showed that moms who consumed cannabis throughout gestation or pregnancy were more likely to have kids diagnosed on the spectrum. And so that's a pretty important key and something worth examining. And so that's kind of the goal of my social interaction in these animals, is being able to kind of not only analyze like how it affects, but look at different components of kind of a bigger picture. Because sociability, like when you think about social interaction, I'm going to go on a tangent here for a second,
Go
okay?
for
But
it.
like
Let's roll with it.
when you think about social interaction, like we as people interact so much, and like animals interact so much, and so there's so much that like we should be able to squeeze from that. And when it comes to social interaction, it's pretty cut and dry in the basic science world. Like it's kind of like, okay, here's a sociability box. Let's toss this animal in there and just like see if they go and hang out with this familiar rat or this naive rat that they haven't met before. You know, like it's just like, okay, sweet. So you time and you look at how much time they spend with each one and done. And
Interesting.
maybe
Okay.
how much it locomotes around like all three boxes. But like, come on, what is that? Like I guess that's the equivalent of like putting me in a room and like putting you in one room and then like some other person in the other. But like, I don't know, you're a pretty interesting person. So like I might spend more time with you even if you were familiar to me versus somebody else.
Maybe, but you got to start the research somewhere. So it is good that even these studies are done, even though it's
Oh,
simple, even though
a
it's got a
hundred
simple design.
100%, because it provides the baseline, right? I'm
Yeah,
not
yeah.
like trashing
No,
them. It's just like,
you want to extrapolate upon it. You want to add to it.
exactly. And
Yeah.
so much. I want to add so much, because rodents do some crazy things. And no, it may not be the same as how we interact, but like it's equally valuable. And we need to kind of glean as much as we can from these animals if we're using them, because you know that's a big cost, and not like just in money wise.
No, no, not at all. And are you trying to develop an SOP for this, for like a new type of social interaction study? Or are you pulling it from another paper? What's your plan there?
So, hoping to use some machine learning techniques that
Oh
are being
wow.
developed in the
Whoa.
lab right now. So, shout out to Howland Lab. It's pretty cool what's going on there. And yeah, we're looking to utilize some of these machine learning techniques to glean and be able to measure some of these like identified, sniffing or like
Yeah.
just like over under just kind of interactions. If you're familiar with rats at all, most viewers may not be. That's cool and more normal. Maybe not in the science world, but like more normal for the average person. But yeah, it's really cool. Like you identify a certain behavior and then you like toss it into this program and then it just like it starts to pick that out of the video, and that opens like so many doors for assessment of
That's
animal
crazy.
behavior that we've never really had before.
Is it like what's the name? What can you disclose the name of the program that you guys use from this machine learning? Is that or you can't?
Yeah, no, that's a good
If
question.
you if you if you feel like you're not sure, then just don't. But I'm just curious, honestly.
Yeah, I might, I think, well, no, I might keep that one to myself because I'm
Yeah,
not like,
I'm not
I know
sure
it's,
either. Yeah,
yeah, I'll be safer rather than sorry
yeah, no, I think you
in that
should,
case.
because I
But if,
think either it's a program that is like available to buy online, kind of like GraphPad Prism, or it's some
mhm.
experimental program that a research group has coded themselves, and it might be the case, and it might not even be published, so that's fair. I was just
That's the thing is
curious.
like I know like Tim Afrenia Offercheck, oh my gosh, is trying to publish on this one. So I don't
Oh.
want to like pull the rug out and be like, psych.
No, I'm just
Um,
that's really cool though. I mean, it's and I think what you're saying here speaks to a cool idea that when you do science, you end up collaborating with a lot of different groups because they have some very specific fine-tuned technique that otherwise would have no application, right? And I think that's a really cool thing about research.
it's so cool. Like it's so cool. And I love the people that do it. Like, man, there are so many cool people out there doing research that are so passionate about it, and like the way that everybody's brain works in their certain areas is amazing. It's truly just amazing. I remember going to that conference with you when we were in Spain and
Yeah,
just being like totally amazed by everybody who was there, and I was like, wow, this has reinstilled my faith in humanity to like figure out the world's problems.
yeah. Yeah, it speaks to that idea that you can like specializing in something versus knowing a lot of general stuff about something, which is kind of the world we've been in the past 100 or 200 years, where we're like specialized really deep in one area so that we can learn a lot collectively as a species. And I think that's just like it's great. It does wonders for the world,
Well,
you
and it,
know.
it gives like a platform, and like you and I are doing right now. Like there's now platforms where like the connection can happen so much faster. Like think about the letters Charles Darwin sent,
I know.
and like how slow that must have been, and being like, man, I hope he got that letter. Like, and
Seriously. You have to
it's just like,
receive, it takes you long to receive the letter. You have to handwrite, a response, mail it back, and then there's that travel time all the way back. Now it's instantaneous, it's
it
like shoot an email. Oh, this person does this. Hi, how are you? Get him on a phone call 2 days later. you know
I know. And like the thing about it is like because people are so specialized, when somebody takes interest in what they do, oh my gosh, people want to share that. Like everybody wants to share what they're doing because like it's really cool, but it's a lot if you don't have a grasp on it. So like I love reaching out to scientists because everyone is like for the most part so friendly and is just like wants to teach and wants to learn. And that's it's just really cool.
it is cool. Yeah. And I think honestly, I think that's where we'll kind of end it for now. I
Okay,
think
chill.
I want to thank you for coming on, and also what we should do is, what exact part in the project are you? Are you breeding right now?
Yes, I am breeding and treating right now. So I'm kind of my
Ah,
first wave of it. I'm in my first
hmm.
cohort. Mhm.
We should. I want to check in with you every sort of when you're done with sort of a phase and ask you how it went. That would be fun to check in
I
with
would
you.
love that. It would keep me on it too. I'd be like, yeah, we've got to get pump this research out so
We've
we
got to
can
get
talk
back
to
on.
Asher's
Get back
following about
on.
it.
Yeah. Yeah, my platform is much more psychedelic oriented, but LSD and psychedelics. But I think cannabis is cool too. Drugs are interesting, right? Let's just I want to talk about drugs.
I know. Well, and like, man, you know how interested I am in all things psychedelics, but like
Yeah,
sometimes you
yeah,
just have
yeah.
to go where, funding and opportunities are, right?
Right,
Like,
right.
what you started out with your PhD. So it's just like,
Yeah.
There's common interests everywhere. Asher, can I plug myself before
Yeah, totally.
before we go?
Tell
Okay,
us tell
chill.
us your your media stuff.
Okay, well, if you are interested in following me on Instagram or Twitter, I'm at Talpaca13, as in like if you want to go for a lunch, alpaca one. And then the number 13. And then I have a podcast, as we talked about, with my friend Elsa, and it's called At Knowledge by Proxy, and we're on Spotify. So if you want to come and listen. But Asher, thanks for this. This is rad.